Welcome to my channel. So this video will talk about the alternate complement system, the lectin pathway, and the functions of the complement. We'll have a quick recap of the last video, that's the classical complement pathway. So first you need an antigen, then you have an antibody. Once they form an antigen-antibody complex, the C1 complement is activated. Now C1 has three parts. It has a C1R, C1Q, and C1S. So in the presence of calcium, the C1S will activate the C4 molecule. The C4 molecule will split up into C4A and a bigger fragment C4B. Now this C4B combines with the previous C1 and forms C14B. Now this will go on the next step of the cascade, which is activation of C2 molecule. Again, it will split up into a bigger fragment C2A and smaller fragment C2B. Now this C2A combines with the previous molecule and forms a C3 convertase, the first important step in the classical complement pathway. So next is the activation of C3, where you have the bigger fragment C3B and the smaller fragment C3A. This is the C5 convertase. Once a C5 convertase is formed, it will activate C5. So C5 will again split up into a bigger fragment C5B and a smaller fragment C5A. Now this C5B along with the C5 convertase is now joined by C6 and C7 initially, followed by attachment of C8 and C9. So this whole thing, that is from C1 to C9 together, is the membrane attack complex. Now this membrane attack complex will now attack on the antigen and form pores leading to antigen killing. Now the smaller molecules, you have the C4A, C2B, C3A and C5A. These are all anaphylatoxins which will enter the bloodstream. So this was basically the classical complement pathway. Now we'll continue with the new one, which is the alternate complement pathway. Now this pathway is completely independent of an antibody. So here the activation happens when via some of the other antigens, for example, endotoxin produced by the bacteria, lipopolysaccharide from the gram-negative cell wall, teichoic acid from the gram-positive cell wall, fungal cells and virus-infected cells. Now, since this is not dependent on the antibody, there is no involvement of C1, C4 and C2. Instead, there will be factor B and factor D and Properdin, which are there in the bloodstream, which will act as instead of the C1, C4 and C2. Okay, so any one of these antigens can activate this particular system. Now the factor B and factor T are serine protease imogens. Properdin is there, and you have the C3, the molecule which is there in the bloodstream. So what will happen? It will keep on hydrolyzing by itself. So it will form the C3A and C3B. Now C3A will again go back into circulation. So the C3B will be there. Now whenever there is a antigen, maybe an endotoxin or a cell wall or infected cell, what will happen is C3B will attach to the antigen. Now there's a combination of antigen and C3B. So next step, what happens? The factor B, which is there in the system, will come and attach to this particular coated part of the antigen. That is, antigen which is coated along with C3B. To this part, the factor D comes and attaches and splits the C3 into C3BA and C3BB. Again, the C3BA will go back into circulation. This is how the initiation of the alternate complement pathway starts. So let's have a look. So you have a spontaneous hydrolysis of C3 into C3A and C3B, wherein this is a free C3. So it will keep on getting hydrolyzed. There what will happen? The C3A will go back into the bloodstream itself. C3B can attach with an antigen, coat it and become a antigen plus C3B complex. So this is now a bound C3 complex. To this complex, the factor B will attach. So now you have a division of the C3A and C3B by the influence of factor D. Once the factor D is there, so this C3B complex is split up into C3BA and C3B, capital B and small b. Now this is basically your C3 convertase. Now this is a very unstable compound. It has a very short half-life. So it has the Properdin system will come and stabilize and increase the half-life of this particular molecule. This C3 convertase now acts on the free C3 in circulation, splits it up into C3A and C3B. And this combination of this C3 convertase with the C3B will lead to formation of what is called as a C5 convertase. Once the C5 convertase is formed, the other steps of the pathway are as same as your classical complement pathway. The third, you have the lectin complement pathway, which is again an antibody independent system. So here what happens, there are lectin proteins in the host immune system that interact with the mannose residues on the surface. Here again C1 is not involved. The other parts, the C2 to C9 are involved here. Okay, so these mannose residues are seen in some Salmonella, Listeria, Neisseria and other bacteria, and also in some places in Candida. So instead of C1, the mannose binding lectin and the mannose binding lectin associated serum protease will act instead of C1. So what happens? The C1, that is your the complex will go and first of all go and activate C4. So C4A will split up as anaphylotoxin. The bigger fragment is C4B. Now this is now acted upon, or this is again activates the C2 complement. Which again the C2B goes off as a anaphylotoxin. So you have C4B2A, which is a C3 convertase in this pathway. Then C3 is converted, so C3A goes out as a anaphylotoxin, and the remaining fragment is your C5 convertase. Now this will activate the C5 molecule, and once the molecule is formed, it will again go and activate C6, C7, C8 and C9, leading to formation of the membrane attack complex. Now this is a complete overview of all three types of systems. Okay, once this particular membrane attack complex is formed, what do? They will basically form larger pores in the pathogen. So what will happen? The ions inside and outside will start exchanging very freely. So in that time, the osmotic stability cannot be maintained. So the bacteria will be killed by either influx of water or loss of electrolytes. Along with this, there are some other actions what happen. First of all, it's an inflammatory response. So you have a C3A, C4A and C5A, which are anaphylotoxins. This will bind to the mast cells and there will be release of histamine, which will cause vasoconstriction and increase vascular permeability. Second thing is opsonization. Opsonization is basic meaning is just like applying soap. So C3B and C4B, they will coat the immune complexes. So once these are coated, the immune complexes, the phagocytic cells will have receptors for these C3B and C4B. So it will be easy for them to identify, and they will initiate the process of phagocytosis. Then the third one is the removal of the immune complexes from the blood, wherein C3B is very important in that. And these immune complexes, that is the infected cells which are coated with this particular component, will be taken up by the system to the liver and spleen, where they are phagocytosed and sent out of the system. Then fourth one comes the viral neutralization, wherein the complements coated on the surface will neutralize the viral infectivity and make the virus particles useless. Then you have the complement receptors, where they have the function of these receptors is the complement mediation and regulation of the pathway. Okay, so the C1, CR1 to CR5 are there in various types of cells, and they will bind to the specific ligands to mediate specific functions. For example, CR2, which is present on B cells, will be involved in the humoral immune response. For this can also act as a receptor for attachment to Epstein-Barr virus. Now, when these complements are active, there are some systems by the bacteria which can also evade this complement system attack. The most common way of preventing this action of complement is by preventing the membrane attack complex insertion. So that can happen in different ways in different bacteria. So for example, E. coli and Salmonella will prevent the membrane attack complex insertion by having a long polysaccharide chain. Same way in gram positives, the thicker peptide glycol cell wall will prevent this insertion. Sometimes there can be the non-covalent interactions, like in case of Neisseria, which will prevent this particular attachment to the cell wall. Then you have the bacterial capsule, which will prevent physical barrier. So it will prevent the particular interaction between the membrane attack complex and the cell wall. Pseudomonas has some enzymes called elastases, which can destroy the C3A and C5A. And some of the viruses and candida have proteins which mimic the complement regulatory protein, so they can regulate and stop this complement action itself. So how the regulation happens? So it's a antigen non-specific. So it is capable of attacking the microorganisms as well as host cells. So it has to be a very specific regulatory mechanism. There are series of regulatory proteins, of which the most important one is a C1 inhibitor, which will inhibit the C1q itself. So what happens? The whole classical pathway itself is inhibited. Then you have the decay accelerating factor, which is CD55 molecule present on the cell membrane, which will basically dissociate the C3 convertase. So all three pathways are completely inhibited. So what are the complement deficiencies, or what are the effects of these complement deficiencies? So if there's a complement deficiency of C1, C2, C3, C4, the classical pathway is affected, and if C3 is affected, it's a common deficiency, so all three pathways will be involved. So in this case, the patient can suffer with systemic lupus erythematosus, glomerulonephritis, and pyogenic infections. If there's deficiency of Properdin or factor D, alternate pathway is affected, leading to increased incidence of Neisseria and pyogenic infections. If the membrane attack complex C5 to C9 is deficient, there's a common deficiency, means all three pathways will be affected, and there will be a disseminated Neisseria infection. Now, when there's a complement deficiency of the regulatory proteins like C1 esterase inhibitor, what will happen? There will be a classical pathway will act in a hyperactive manner, leading to hereditary angioneurotic edema. If there is a decay accelerating factor, that is your CD55 is deficient, what will happen? There will be a deregulated C3 convertase, which increases RBC lysis. So that can cause paroxysmal nocturnal hemoglobinuria. So this is the overall presentation of your complement system. So those who have not have the time to go to the previous video on classical complement pathway, please do watch that video. I hope you like this video. Like and subscribe to my channel, and share this video to those who might be useful for this. Thank you.